Hello, everyone. Welcome to today's live broadcast, Applying ISO 10993 Part 18 on Extractables and Leachables Characterization, Supporting the Medical Device Regulatory Pathway. I'm Laura Bush, the Editorial Director of LCGC, and I'll be your moderator for today's event. We're pleased to bring you this webcast presented by LCGC and sponsored by Waters Corporation. Waters Corporation, a world-leading specialty measurement company, has pioneered chromatography, mass spectrometry, and thermal analysis innovations, serving the life, materials, and food sciences for more than 60 years. With approximately 7,200 employees worldwide, Waters operates directly in 35 countries, including 15 manufacturing facilities, and with products available in more than 100 countries. Before we get started today, we have a few housekeeping announcements. First, the webcast is designed to be interactive, so we encourage you to ask questions. You can submit your questions at any time by typing them in the Q&A box, and you can find that on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. And if you have any technical problems viewing or hearing the presentation today, please click on the question mark Help widget in the dock at the bottom of your window. I would now like to introduce today's speakers. We are very pleased to be joined today by Rachel Sanig and Ben McCreath. Rachel Sanig received a first-class master's and BSc honors degrees in chemistry from the University of Leeds, and she is a member of the Royal Society of Chemistry. She joined Waters as an applications scientist in 2016 and is experienced across the clinical, pharmaceutical, and biopharmaceutical areas using a range of LC-MS analysis techniques. She joined the Chemicals and Materials team in 2018, where she works in the areas of fine specialty chemicals and materials research, with a focus on applications in extractables and leachables analysis. Ben McCreath completed his PhD in polymer synthesis after a master's degree in medicinal chemistry. He has spent most of his career working with instrument vendors at the interface with the markets they serve, from the science and application development to developing software chemistries and instruments. His current focus is around understanding and working with industry and academia to develop better solutions for their requirements, and on providing solutions for the chemical and material sciences through the Waters and TA Instruments portfolios. Thank you both for joining us today. Ben, please get us started.
Thank you, Laura. And thank you, everyone, for joining us today for a talk on extractables and leachables for medical devices. So this is a critical part of the ISO 10993 standard, specifically covered within Part 18, chemical characterization. As Laura said, I'm Ben McCreath, responsible for the medical device market at Waters. The main speaker today after my introduction will be Rachel Sanig. Today we're going to take you through the extractables and leachables within ISO 10993-18 for the analysis of medical devices. We'll highlight how some key technology, such as HRMS, helps users meet the guidelines of the ISO, as well as some key functionality and parameters that help users deploy this. This can be quite an advanced analytical application. We're going to help you see how to simplify the deployment into your laboratory with confidence. And fundamental to the success is how an analyst can encompass all of this within a workflow-based application. So the suite of standards known as ISO 10993, the biological evaluation of medical devices, has seen major changes to implementation over the past few years, including Part 1 in 2018, Part 18 at the start of this year, and no doubt more to come. It encourages a risk management approach. So to perform the biological evaluation of the device, your device's intended use guides the line of thinking, the nature of the body contact. Is it in contact with the external skin, or is it implanted? You also need to consider the contact duration. So is it only used for an hour, or can it maintain contact for weeks? So historically, this is used to dictate the biological testing. Now the standard appreciates that if you know the material formulation, if you understand the chemistry, then you can predict the risks. So it's inserted physical and chemical information as the key step to intelligently design a biological evaluation plan. Therefore, having a strategy to build up your understanding of the chemical and material nature of your device is key to a smoother process through the regulations. There are many parts to ISO 10993, but the chemical characterization, here are some of the most pertinent ones, ignoring the degradation products for now. So Part 1 describes what is a medical device. Part 12 describes extraction conditions, so using temperatures, time, solvents. We would also be referring to Part 17, which describes how limits for chemicals that might leach out of the compound to the patient, so the leachables, how they are set. Part 18 describes the analytical techniques that can be used to understand the chemical and material properties. So today's talk will be focused on the extractables and leachables components. We're going to focus on how E&L can be implemented to achieve this. For more detail on the regulations themselves, I would point you to an earlier webinar in June presented by Dr. Andrew Fielden. The ISO 10993 Part 18 provides a workflow for how to approach this study. This is reproduced here. The first step is to gather information on the device, its intended use, and the materials and chemical constituents. Depending on how much information you have from your suppliers and the device's intended use, it's possible to come to a decision on biological testing at this point. For example, if there's already a substantial equivalent product on the market and the device itself is classed as low risk. However, if there's not enough information or the risk is not acceptable, then further testing is required. This is conducted through extractable and leachable testing, and this is what Rachel will be talking to today. There are a few terms in here which, although may be well known by the audience, we'll quickly cover now. Extractables are the potential impact of the device. Conditions are used that are beyond what the device is expected to experience to draw the chemical moieties out of the material. These are known as exhaustive or exaggerated conditions. Alongside extractables, we have leachables. These represent the actual impact, what substances are released from material during its clinical use. It's very difficult to replicate the actual conditions, and so simulated extraction is used to produce a profile that represents the worst-case leachables profile. These terms are defined in Part 12 of the ISO 10993, and this should be consulted when developing the sample preparation methodologies. We won't be going into depth on these today. The potential chemistries within a given product are vast, necessitating a range of techniques to cover the full range of chemistries. We're focusing on the non-volatile organic compounds today, with liquid chromatography as a separation mode. There could well be a significant quantity of moieties that can be detected, especially when extremely sensitive technology such as mass spectrometry is employed, and not all are going to be at the level of concern. So to this end, the term known as the analytical evaluation threshold is used. Now this is a very important concept within ISO 10993 Part 18. Below the calculated threshold, any extractables or leachables need not be identified nor quantified. This means, of course, that anything above this threshold, you need to identify the substance, you need to quantify the substance. You need to work with the toxicologist to understand what levels of compound are important. So if you think about this from an analyst perspective, you start with an extractable study, determine the AET, and perform the extraction according to the conditions appropriate for the device. Using a screening methodology to collect the data, in this case we'll be using LC-MS, then apply the AET, the threshold which dictates identification and quantification of peaks. For anything above AET, the analyst has to identify, having a list of potential candidates from the supply chain, which greatly simplify this process. If it is a complete unknown, then elucidation tools are required to confirm the structure. Once this is completed, then it can be quantified. At this point, if a decision can be made, you can finish. If, however, further information would be useful, what is the quantity likely to come out rather than the more aggressive extraction conditions that we've used for an extractable study, then you have a leachable study. In this, you and your toxicologist select targeted compounds from the extraction study to target. So therefore, the leachable process is more focused. A list of compounds from the extractable studies is identified for targeted quantitation. This has less demands on the technology and software than elucidation studies. To go through this in greater detail, I'll hand over to my colleague, Rachel. Over to you, Rachel.
Thank you, Ben, for that overview. I'm going to now talk to you today about how Waters can help in extractables and leachables testing for medical devices. If we think about the manufacturing process of plastics, most commonly used material in packaging and devices, there are several steps along the way from the monomer to the final product. Each of these steps can introduce additional impurities and degradation products, and manufacturers might not be aware of the impurities in their raw materials, or they might just not disclose this information. So we don't always know what's there that we're looking for. Here we have summarized some of the most typical extractables. we've got impurities, chemical additives, monomers and oligomers, volatile compounds, and residual processing compounds. So what we can be looking at are very complex materials. As Ben touched upon, there's a range of analytical techniques needed to cover these typical extractables. For example, to identify volatile organic compounds, you would opt for gas chromatography, mass spectrometry, maybe using the headspace technique. Electron impact or chemical ionization can be used here. For semi-volatile organic compounds, you would most likely investigate these also with GCMS, and atmospheric pressure gas chromatography can also be used. For those non-volatile organic compounds, and some semi-volatile, liquid chromatography mass spectrometry is the go-to technique, and this can be used with electrospray or atmospheric pressure chemical ionization. For anions and metals and elemental impurities, ion chromatography is needed, or inductively coupled plasma mass spectrometry. At Waters, we can supply a separation solution technique for different analytes depending on their volatility. For those non-volatiles, we offer UPLC, Ultra Performance Liquid Chromatography, with an electrospray ion source. UPLC means we can operate at higher pressures with low dispersion, getting sharper peaks with increased throughput. Here we can see our Acuity UPLC ICAS system. We have a binary solvent manager, sample manager, and column heater within one stack. We also have a PDA in line here, so we can include UV tracers if we need. For semi-volatiles, we offer gas chromatography with atmospheric pressure gas chromatography ion source. This combination provides GC users with a soft ionization technique that increases sensitivity and specificity. Using the Waters universal source door design, we can switch between the techniques on the same platform with minimum downtime. I'm going to focus on extractables and leachables analysis using LCMS moving forward. At Waters, we have a focused and well-integrated end-to-end solution in this application area that encompasses our core competencies in chemistry, separations, mass spectrometry, informatics, reagents, and standards. It's supported by a global service and application support capability, ensuring customer satisfaction and success. But during my part of the talk today, I'm going to give an introduction to some of the key technologies, explaining how they help overcome the challenges customers encounter in E&L analyses, followed by how we can use an application-specific software to take analysts through the screening of medical devices. So here are just some of the challenges for screening of extractables and leachables. You need to separate and detect all components, and we want to be able to identify the compounds present above the AET level, whilst trying to avoid false positives. And you want to get as much information about the components as possible to help with elucidating any unknowns. I'm going to describe how our technologies can help to combat these challenges. When running our screening analysis, we want to be able to find as much information about compounds that are in our sample as possible. This helps to identify what the compounds are, be that through matching against the library or through structural elucidation. Here we can see the mass spectrum for Ergonox 245, a common antioxidant. On the top, we can see the low energy spectrum containing the precursor ion. Below it is the high collision energy spectrum, containing the fragment ions associated with the precursor. Using high-resolution mass spectrometry, HRMS, and a capability called MS3E, in one analytical run we can simultaneously obtain both the accurate mass spectrum of the precursor and the fragment ions, providing us with information that we can use to both screen against the library and to help with elucidation of a structure. Here is the schematic of the Waters Xevo G2XS QTOF. The excess collision cell, which is highlighted in red in the middle of the schematic, is what lends us the capability to run MS to the E. Alternating low collision and high collision energy is applied, enabling the simultaneous acquisition of precursor and fragment ions throughout the entire chromatographic run. Later, during the data processing, precursor and fragment ions will be precisely aligned based on their retention time. So for every peak, we will have the exact mass of the compound and its corresponding fragments. And as I said, this information helps when we're screening and in structural elucidation of compounds. So whilst many of you are probably familiar with the use of nominal mass and tandem quad technology for screening, high-resolution mass spectrometry, HRMS, using both quadrupole and time-of-flight mass spectrometry is becoming increasingly attractive. For targeted screening, its sensitivity now rivals tandem quad systems with better specificity. But more importantly, not only do we have the ability to screen for an unlimited number of targeted compounds, we can also get very good information about all our peaks found in our samples, helping with the structural elucidation of unknowns. The data acquisition is easy to set up, and the data can be investigated retrospectively. Typically, when running LC-MS screening experiments, you want to start with some kind of user library, so that when we screen our samples, we can match against the library to identify compounds. We need to think about what information should be included in such a library to help this process. To start with, you would usually populate a library with retention times that's based on a single method. Next, it helps to also include the accurate mass of the compound. When we screen using high-resolution mass spectrometry, we can get high mass accuracy, which helps to match our compound identity with the library. In some cases, information of fragmentation is available. And again, with MSDE scans, we can find fragment ions for all our precursors, helping with library matching. When you have all this information in your library, how confident are you that you have the right matches? In cases of large targeted screening lists, it's useful to have as many identification points as possible to avoid false positives. There are some challenges associated with the information that we have here. For example, retention times can shift with new mobile phases or a change in column or across different instrumentation in different labs. Typically, ENL analysis is done by a generic gradient. Therefore, conditions might not be optimized for every precursor, resulting in an absence of fragments. Also, what if you have isomers present? The accurate mass as an identification point is not valid for such cases. So, we need to think about what additional information could help us in this case. This is where we can turn to a value called the collisional cross-section of an ion, the CCS value. The CCS values can be obtained by a technique called ion mobility spectrometry. It can be advantageous as an additional identification point, helping to reduce false positives when screening. Unlike retention times, CCS values are not affected by changes to mobile phase or by column or by matrix effects. Furthermore, where accurate mass alone would fail to separate isomers, CCS values could potentially be used to separate and identify any isomers present. Therefore, the additional CCS values to the extractables and leachables screening library can help aid successful targeted screening. So, what are CCS values and how do we obtain them? CCS values, as I said, are found by using ion mobility spectrometry. At Waters, we have the Vion IMS QTOF. And here, in addition to the collision cell that you'd see on the Xevo system, the Vion has an ion mobility drift cell, labeled mobility separation here. Ion mobility spectrometry, IMS, coupled with HRMS, affords an additional separation of ions based on size, shape, and charge as they drift through gas in the mobility cell. The collisional cross-section, the CCS, value of an ion may be determined using its drift time through an IMS device. CCS is a distinguishing characteristic of an ion in the gas phase, related to its chemical structure and three-dimensional conformation. You can see the CCS represented here by the ion trapped within a sphere. Its CCS is the shadow cast by that sphere. Under typical low energy operating conditions, CCS can be a robust, precise, and measurable property of a variety of ionized species. And as I mentioned, it can be beneficial to include in our screening libraries as an additional data point. Furthermore, to the benefits we get with CCS values, we are also afforded an additional degree of separation when using ion mobility. Here we can see two co-eluting compounds that have not been chromatographically separated, but can be separated by drift in the mobility drift cell. They then get separated by mass to charge, and what results is drift time-aligned precursor and product ions. In this example here, we can see two compounds, phosphate and Ergonox 245, that co-elute with a retention time around 7.48 minutes. Without ion mobility, we get this mixed mass spectra for both precursor and fragments, which can make elucidation quite challenging. When we use IMS, we can see the benefits of the additional separation, as we're able to separate the two compounds by their drift time, and we can get separate spectra of the precursor and fragment ions of these compounds. Another advantage of IMS separation is that we can get increased spectral clarity when components are aligned by their retention time and ion mobility drift time, making easier to visualize and identify those precursor and fragment ions. Low and high energy spectra become cleaner as chromatographically coeluting species with different ion mobility drift times are removed, and fragment ions are more easily associated with their precursor ions as their drift times align in the ion mobility dimension. Univol 330 shown here, you can see on the left that the spectra without drift time alignment and the spectra on the right with drift time alignment, and you can see that the spectra becomes a lot clearer. So in addition to our high-resolution mass spectrometry platform, the Xevo G2XS QTOF, we also have the Vion IMS QTOF, which allows customers to access the benefits of ion mobility base separation and measurement, which helps towards clarity of spectra and confidence in screening results. So hopefully I've demonstrated here the benefits of HRMS and IMS instruments in the screening analysis of ENL, helping to improve separation and detection while also providing us with more information to aid successful screening and elucidation of unknowns. Both these platforms are fit for purpose for this type of application, and they can address some of the challenges faced when analysing these very complex devices. So I've talked a lot about the benefits of HRMS and IMS, but where does this fit into our ENL analysis of medical devices? As Ben discussed earlier, we need to answer the following questions as an analyst. We want to screen the compounds in our sample of interest and determine what's above the AET level. We want to identify those compounds either against the library or through elucidating the unknowns. After this, we'd want to do quantification as well. With HRMS and MS to the E capability, we get the accurate mass of precursor and fragment ions simultaneously, which is very useful when dealing with those very complex materials for medical devices. It helps with screening against the library if we can match accurate mass and fragment ions, while also providing key information to help speed up the process of elucidating any unknowns. With ion mobility, we get that additional degree of separation, helping to reduce the complexity of our spectra and pick out components of interest. Using IMS, we can potentially reduce the number of peaks above the AT that need characterization, as the sum of two coeluting peaks may exceed the AET, while separately they could be below. Separating those two peaks up front could avoid unnecessary work, and alongside cleaner spectra, this makes identification of components much easier for the user. As I've mentioned, having CCS values gives that additional identification point in our libraries, helping to reduce the chances of false positives and increasing our confidence in the identifications. For our targeted screening of potential leachables, both HRMS and IMS benefits can be utilized. However, if you're looking to routinely run targeted leachables screening, then nominal mass techniques would work well here. So now that we have our instrumentation for the ENL analysis of medical devices, we need to look at the software side of things and see how we can progress through this complex workflow. At Waters, the software we use is called Unify. It's a single software platform for chromatography, mass spectrometry, data management, and lab workflow. It encompasses data acquisition, processing, visualization, and reporting. It's based on a database structure with configurable compliance tools. So the software can be set up in a fully compliant mode if your lab environment requires so. It is an application-centric workflow, and as I'll show you in the next few slides, the workflow can be easily customized to meet specific ENL needs. Let's start by looking at the steps that encompass our Unify workflow and how we get our system ready for the data analysis. We begin with a system setup. On the Vion here, we can see a wizard-based automatic instrument setup to get the HRMS and IMS ready in a time-efficient manner, calibrating the mass accuracy and the CCS. When the instrument is ready, and we have extracted our samples in accordance with the ISO recommendations, we can now select the analytical LC-MS method for our injected samples. Here is an example method that how we run ENL type analyses here at Waters. You can see our typical LC conditions on the left and our typical MS conditions on the right. We also want to set up a library associated with the run, the database of compounds that contains a list of typical extractable and leachable analytes. You can see here an example of Ergonoc 1098 in our Unify scientific library. For each compound, the library contains critical information such as chemical formula, structure, adduct ions, and the CCS values for each of those, precursors, fragment ions, retention times. As I have explained, the use of multiple identification parameters improves confidence in the identifications made. Authors can provide customers with a large E&L library that we have developed as part of our solution, but customers can create their own Unify libraries very easily after acquiring and processing data with standards. So we've extracted our samples and set up our screening method and library, and we can now inject our samples and acquire our data in the MS to the E mode so that we can simultaneously acquire those precursor and fragment ions. Once the data has been acquired, it is processed by the software, and then we can move on to reviewing the data. And this is where we can look to answer our questions. What has been identified above our AT against the library, and if there are other unknown compounds that are there, we need to elucidate them. And how much of these compounds are there present? Finally, when the data review is completed, Unify allows us to generate fully customized, flexible E&L reports. I'm going to talk through the data review stage in more detail and how we can use workflow steps to reduce the complexity. I'm going to show some examples from different E&L analyses that we've run in-house at Waters. So here we can see how the Unify workflow can be set up to fit medical device extractables analysis. The workflow can be designed by the user and is fully customizable. It is simple to set up and helps to organize and structure a complex HRMS dataset within a screening application. So on the left here, we will always be able to see the workflow steps that we have customized in Unify so we can easily maneuver through the data. First thing we want to do after we have acquired our data is to check the results of a test mix, a system suitability standard, to ensure the system is performing well. The standard used here is the Waters SST mix, which contains 18 compounds representative of common industry polymer additives and preservatives, which covers a wide mass range and includes compounds that ionize in both negative and positive ESI modes. There are different ways to view the data in Unify. For example, here we can look at it as a component overview plot, which gives a visual representation of the components of the standard that have been identified, and we can see their accurate masses and the structure assigned by the software. Another way to review the data is through the component summary table of identified compounds. It contains information such as retention time, mass error, and isotope matching parameters, which can be used to verify instrument performance. And we want to see things like retention time reducibility and good mass accuracy. Within the same screen, you can view the chromatogram for all identified components and the chromatogram of just the selected component. In this case here, Univol 330. We can also see the high and the low energy mass spectrum that are associated with it. We're also able to see the chemical structures that have been assigned to the precursor and fragment ions if you were to hover over these little blue symbols in the software. This helps to reduce manual interpretation. These views are all customizable, and you can display the data as you want to see it. We can also change to a summary plot view. I like to use this to visualize the analytes of interest across all injections. In the case of the system suitability test, we can check we are getting reproducible retention times. Here you can see the retention time of Univol 330 across 3 injections of our standard mix. And we can view trends for any of the data that we've collected for our component. The next step is to review the screening results of the samples, and we can get a summary of everything that has been found identified in the sample at all levels. But for medical devices, we are only interested in the components that lie above the AET. To view the components at certain levels, we can set filters in any workflow steps to filter out the components that we are not interested in. Again, we can review the results in an overview plot or a component summary. We can see the components, the screening analysis identified against the library, and all the associated data with each component. Highlighted here, we can see ErgoPhos 168 and its chromatogram and spectrum, and all the data that is collected. So as mentioned before, we can view different trends, so we can see what compounds are present in our sample across all injections. Here, for example, I've sonicated a PLA sample and 3 different solvents for 8 hours. And if you look along the bottom of the summary plot table, you can see the name of each injection. We can see that ergophos 168 has been identified in the vials that contain IPA and hexane, but without any of the PLA sample. But they are highly elevated in the ones with the PLA, especially when using IPA solvent. This helps to give us a good overview of how we're seeing our compound across all injections. We can also track across samples using CCS values. Here you can see that ergophos 168 is picked up in the injections as it is expected to. It gives us an extra layer of confidence that the identifications of this compound across all the injections is accurate. As mentioned before, we also want to be able to quantify those compounds that are above the AET, and we can build in analysis calibration curves for compounds in our targeted library list. Here you can see at the top of the window the calibration table with 4 calibrants at different concentrations. At the bottom, you can see the chromatogram of each calibrant, the integration of each peak, and the calibration curve built from them. If we haven't been able to set up a calibration curve, and if we've used an internal standard, we can include response factors in our component summary table, which will give us a first look at the estimated concentration value. So after we've built our calibration curve, the next step is to calculate the concentration of each sample based on its signal. The example here shows the quantitation results of packaging samples that my colleague has extracted. At the bottom, there's a summary plot. The blue bar represents the concentration of the targeted analyte in each of those 6 samples. The summary plot again allows you to easily compare samples and see how the concentration varies in the whole batch. So after we've screened against our library, we want to know what else is in the sample above the AET level. The identification of unknown peaks in the UPLC chromatogram is a critical part of the analysis of ENL to achieve full characterization of the sample. The software starts by finding and listing the unknown compounds, and again we can filter to only look at what is found above the AET. The next step is to use comparison tools. Here we are highlighting a feature in Unify called Binary Compare, which allows the analyst to directly compare results of an analyte sample and a reference blank extract and identify key differences. This is a really important tool for ENL analyses, because background levels of ENL type compounds are often seen. We highlight Binary Compare here, but there's a multivariate analysis suite that we can also utilize if we need statistical comparison. So, back to our Binary Compare, the sample here is shown in the blue trace, and the blank is shown in the red trace. And if we combine these together, we get this difference plot, which is shown in green. And this shows what is unique or elevated in our sample compared to the blank. If any of these peaks are above the AET, they will need to be fully characterized, and those components that are unique to the sample or highly elevated can now be easily displayed in the component summary for further review. Here we can see in the window at the top a list of unknowns that need characterization. We can see their chromatograms, we'd also be able to see their spectra, and here we have a pseudo trend plot highlighted for them. We can select a candidate mass that we want to investigate and take it straight from the workflow into Unify's elucidation toolkit. So here we can see the Unify toolset, where we can utilize the discovery tool to investigate components. Once you've filled in some specifications, Unify runs the discovery tool once you hit start. The tool combines 3 important functionalities: elemental composition, on or offline database searching, and fragment matching. The toolset starts with elemental composition calculator that determines the most likely chemical formulas for an accurate mass peak. An IFIT algorithm is used to score each formula by the likelihood that the theoretical isotope pattern of the formula matches a cluster of peaks in the spectrum, along with the high-resolution accurate mass measurement. The software then uses the predicted elemental composition to undertake database searches for structures, for example in ChemSpider. Next, it will do an in silico fragmentation of the found structure to look for matches of the theoretical fragments with the observed high-energy ions. Tentative results then get displayed for the user, and you can see here it's given a structure of the proposed compound alongside that measured high-energy spectrum with assignments of fragments of the proposed structure. You can also go directly to view the compound in the database where it found this structure. To get a 100% match, a reference standard would be needed. However, the Unify software saves hugely on the analyst's time in the elucidation process. Once you are confident in the identity of the component, this compound can then be added into our original library so that when we next come to screen a medical device sample, we will have this in our library to screen for. So today I've talked through the benefits of using high-resolution mass spectrometry and ion mobility in a screening analysis, as well as how an application-focused software workflow is beneficial to an analyst in the analysis of extractables and leachables of a medical device. There are some other elements that we offer at Waters that can benefit this process. Our chemistry team has developed and optimized a 15-minute long generic LC screening method using an Acuity Cortex C18 column that offers the highest separation capacity. We also have a unified E&L library that has been developed and is available to all of our users. It currently contains over 700 compounds, with many entries containing fragment mass information, retention time, and CCS values. While we're continuing to expand our own library and build on the quality, as said previously, customers can easily develop their own libraries on top of it. E&L testing is a challenging application, partly because polymers are everywhere. Consequently, particular care should be taken for sample and system preparation. We recommend the use of our certified vials and a specific system cleaning protocol. We also have a system suitability test mixture that I mentioned that contains a mix of 18 common polymer additives. This should be used routinely to check the readiness of the system before analysis of real samples. And this is also helpful in case of troubleshooting. Last, but certainly not least, in addition to all those tools, our application chemists have plenty of expertise and experience in this area, and they continuously train and support our customers to use our total solution in the most effective manner. So with that, I'm done with my part of the presentation. Thank you, Ben.
Thank you, Rachel. Extractables and leachables within ISO 10993 Part 18 starts with understanding what is in your device above the analytical evaluation threshold. With no unknowns being allowed, elucidation is a critical part of this process. Hopefully you've seen today how TOF technology and iMobility can help answer this requirement. Once elucidated, the more simplified mass spec technology can be applied for easier deployment in your workflow. Waters has tandem and single quad solutions for this purpose. The vast potential range of chemistries involved means that the techniques and technology used to isolate and detect can be different. Waters has a universal architecture system which facilitates swapping of the sources on the mass spectrometers, maximizing investment made and enabling incredibly chemical coverage. And although we didn't cover the separation part today, we have separation platforms that are designed to cover the application space to suit your needs. In particular, if you just need to do the routine targeted screening with leachables, so there are no longer any unknowns, you are simply targeting certain compounds in an acquisition method. And there's a range of quadrupole mass spectrometry systems which are easily deployed. With the Acuity QDA shown here on the left, we can enable mass data to be used for detection and confirmation of identity. Here we can see it sat upon top of our UPLC stack. Or we could turn to tandem quadrupole systems such as the Xevo TQ-S Micro, seen here next to the UPLC system, and the TQ-XS. These systems use attention time and monitor the ion ratios of multiple specific precursors to product ion reactions, confidently detect and confirm compounds at the lowest levels. The appropriate solution would depend upon factors such as the sample complexity, level of information required, and detection limits, for example. So to summarize, extractable and leachable testing is a critical component to satisfy the requirements of ISO 10993. Waters has the full solution from the technology to the expertise to address this application. When dealing with complex samples, there will be unknowns that require elucidation, but you need to have confidence that you've captured everything present, and high-resolution mass spectrometry and iMobility are enabling technologies to bring success. To take this technology, this relatively complicated analytical process, and successfully deploy it in your laboratory, potentially in a compliant environment, puts huge demands on the users. Our software is built with workflows in mind, so these can be defined to follow the ISO 10993 Part 18 guidance, facilitating the process and taking more of the burden away from the user. I'd like to thank you for your attention today. We're here to answer any questions you might have on this subject. We'll try to answer as much as we can today. And for those we can't answer or get to, we'll answer by email after the event. Thank you.
Thank you both for those informative presentations. It is now time to begin the question and answer session with the audience. As a reminder to everyone, if you'd like to submit a question, you can do that by typing it in the Q&A box, which you can find on the right-hand side of your presentation window. I'd also like to let everyone know that joining us for the Q&A session is another expert, an additional expert, Caitlin Costa, who's a senior application scientist with Waters, and she may jump in and help answer some questions. Okay, so let's get started. So here's one to begin with. What are the differences between medical device regulations and pharmaceuticals when it comes to extractables and leachables?
Okay, so I'll take that one, I suppose. So, the workflow, once the sample's actually in an LCMS file, is actually pretty similar across those ENR studies. In general, the main differences lie in how the sample gets prepared, the sampling, the conditions of the extractions, and then also the thresholds and the analysis when you come into analyzing the data. But that middle section of the workflow is actually very similar.
Great, thank you. Well, here's another one. Libraries for peak identification are typically very limited. Are there any recommendations from Waters to tackle these unknowns above the AET, which are typically the vast majority of peaks in a chromatogram?
Hi, yeah. So I kind of spoke to this a lot in the presentation. And as I mentioned, we have got a lot of tools in Unify. And because we're collecting MSD data as well with accurate mass data, we've got lots of information about every peak, so including all the ones above the AET that we need to identify. And then when we take this information into our Unify toolkit to do our elucidation, it will search known databases and automatically match these compounds against using a combination of accurate mass and isotope measurements and the presence of those high-energy fragment ions. So that really helps with the confidence in the elucidations that we're making.
Great, thank you. Is it possible to reliably quantify extractables and leachables using a single quadrupole instrument? Do they have enough sensitivity and selectivity for quantification?
Ooh, okay, so I'll jump in on that one. So for what we've discussed today, I'd always stress the need for high resolution MS to get the selectivity, the MSMS to elucidate the unknowns. But once you've fully characterized your material, you might be fortunate enough to be able to deploy a single quad to monitor particular ions of interest. So with selected ion recording, you can focus and improve the sensitivity that you get on a single quad. And of course, with the pseudo calibration, you can quantitate. But yeah, I mean, if the spectrum is still complex, or if you don't still have enough sensitivity, then the tandem quad might be more applicable as well. So you may be able to drop that into your workflow as well.
Can the Unify software only be used with data generated on Waters instruments?
So Unify, it's a really powerful piece of software that we have for data acquisition, processing, and management. And at the moment, we can process both Unify and MassLynx data using the Unify software platform. And we're currently developing a range of APIs, so application programming interfaces, that will enable users to interact with third-party software. So if you want to know about that more, then get in touch with us. You can send us an email with regards to that.
Great. Is Waters specialized? Waters is specialized in medical devices, extractable, and leaches. Sorry, I'm having trouble spitting this one out. Waters is specialized in medical devices, extractables, and leachables, or in other areas as well? Question mark. I'm asking about single-use technology materials, which are very often used in bioprocessing.
Yeah. So I suppose that's again in terms of the E&L process itself. Then this technology can obviously be applied to across the board, across different markets. As long as the extractable and leachable compounds that are expected work by are amenable to LCMS, then our technology will help you answer those. So it's kind of like a similar question to before. It all depends on the different regulations kind of have a different impact on how you treat the sample, but the actual process of detecting and identifying the extractables and leachables themselves is very similar.
Right. Thank you. What is the difference between ISO 10993 Part 18 and USP 1663 and 1664?
Oh, okay, I'll grab that one as well. Sorry, there's a lot of numbers there. The USP 1663 and 1664.
Yes, compared to the ISO 10993 Part 18. Correct.
Again, so this kind of gets down into the detail between them. So the actual differences are very much down at that lower level. So again, it's almost an entire separate webinar to kind of cover those. So what we do have is actually some material. So what may be a good one for this one is that if you pass these questions over to us, we can answer that with a personal email with the attachments to kind of explain that in better detail. It's going to be a difficult one to get across now.
Sure, we can arrange things so that you can follow up online. Fantastic.
It's a very good question.
How are you dealing with the variation in relative response factors when applying the AET?
So that's a very good question. It's a very hot topic at the moment with mass spectrometry ENL experts, and is often discussed at any conferences, ENL medical device conferences that you might attend. It's quite, but some of the things you can use to help with the variation that you get with response factors can be using a range of standards that match your ionization efficiency of the compounds that you're looking for. You can drop your AET level by a certain percentage to help overcome any response factors variation that you might be seeing. It's worth reading a paper that came out recently by Geordie Labs, who they go into a lot of the research they've done on the topic, where they look at using multiple reference standards to help with variation in response factors, and it's something that everyone's very interested in at the moment.
Excellent. So if I'm going to be doing ENL studies with medical devices, where do I start with understanding all the different components that could be in the device?
So Ben, you might be able to help with this, but it helps to do a lot of research into your device. Talk to the.
I think we lost your audio there for a moment, Rachel. Are you there? I'm not hearing you. Okay.
Well, Rachel, guess we can. I'll continue with the answer then. So, it's very important to kind of speak to your supply chain. So the more you can engage and understand from your supply chain, the better position you're going to be in. So that you want as much visibility from the potential components that are in there. So the more you can build up that list of compounds, the more that you can build yourself a library and be more targeted with your methodology. So reduce that amount of elucidation work that you've got to do. And of course, it's important to consider not only the materials but the processes too. So I don't know. For example, polymer devices. You've got all the low molecular weight chemical substances in there. So your catalysts, process aids, you'll have residual monos maybe. But then you've got steps in the manufacturing process for a medical device, which could cause other unintentionally added substances. So stresses from processing or sterilization conditions, cleaning conditions. So it's a good knowledge of material science is going to stand you good stead here. So what we'd expect to come off this device, or once you have this data, you can kind of look at it and go, well, does it make sense? Could this feasibly be present? But yeah, so, I mean, certainly my suggestion would be working with suppliers that have increased transparency. It should be noted that the standards and regulations are also using language to this end as well, because they realise how difficult it is to do this study with blindly on an end product.
Great, thank you. Just confirming we have lost Rachel. I think she's trying to dial back in, but we'll keep going here. And maybe as needed, Caitlin, you can step in and help on some of the questions where you might be able to help. How easy is it to add new compounds to the screening methods?
Hi, everyone. This is Caitlin speaking. Just while Rachel dialing back in. It's very easy to add more compounds to the screening approach. When we're doing the screening approach, we're screening against our Unified Scientific Library. And as Rachel mentioned, that's completely customizable. Also, the workflow approach allows us to go through and elucidate. So once we've done our targeted screening, actually elucidate the unknowns to work out what they are. And once we have our identifications from the unknowns, we can immediately return that data back into our scientific library. So we're continuing to develop and expand that targeted screening library as we analyze samples.
Excellent. And I think we have Rachel back on the line. Rachel, do we have you again?
Hi, can you hear me okay?
Yeah, a little bit breaking up. We'll see how this goes then. So let's try another question. Rachel, this one I think is up your alley. The flexibility of workflows in the scientific library is quite important. Can you explain how you've developed and optimized your LCMS method?
So we've started with different methods in-house that we already use, and we wanted to consolidate these down to have the best optimum method for a generic screening analysis. And that which would then help to improve the quality of our libraries when screening. Of course, we're never going to have the perfect method for every analyte out there, but we wanted to be able to get as much, see as much as we could with our method. So I've analysed lots of different typical polymer standards, a mixture of masses and polarities, look at different mobile phases and things like that. And there were some things that we found that worked better for this type of analysis. So we didn't want to start with too high an organic mobile phase, because then we lose anything that's a bit more polar in the void volume. We also found that you needed to have quite a long hold at the end of the organic mobile phase to try and push off any of those really non-polar typical ENL compounds that you have. And if you don't do that, you tend to see this kind of wraparound effect where they come off in later injections earlier than they should, which can mess with your screening libraries. So there's lots of different things that we took into account when developing our method.
Thank you. Is this the same leachables testing you would use for ISO 18562?
Okay, I'll take that one again. So 18562. Okay, so the ISO 10993 is for the device itself. So what's present within that could feasibly reach the patient. So for breathing apparatus, there's also the gas pathway to consider, which isn't covered by 10993. But it is somewhere where liquid condensate can form and then subsequently transfer to the patient. So ISO 18562 tests the inner gas pathway surface for leachables. So the same principle applies. So you'll need LCMS for the semi to non-volatile samples within there. And again, the kind of the workflows the same in terms of the ENL process itself. Once it's in the vial, the difference is in the sampling and the treatment of the data. So it's not the same, but it's very similar.
Thanks, Ben. How do I go about selecting the right conditions for the extraction?
Welcome to the On24 conference, Rich.
Okay, so So yeah, we didn't really go into any of this detail particularly today, but within Part 18, there's an Annex D, which talks to this point, and you kind of read that in conjunction with Part 12 of the same ISO. And that covers the sample preparation and also talks to extractions, Part 12 does. So in short, the longer the contact time with the patients, like the harsher the conditions required to represent the worst case scenario. So like a long-term device will require 2 extraction solvents of different polarity and an exhaustive extraction. But you know, with such a variety of devices in new cases, it really can't be overly prescriptive. So you need to use the guidelines to logically build a process and then basically justify your procedures. Again, that's actually probably a good subject for a totally different webinar.
Yeah, a lot of these questions have long answers and pretty involved situations to discuss. Here's another one. Would this technique also detect any degradation?
I've got that again. I suppose it depends on the context. So in terms of extractables and leachables, the conditions you use actually must avoid causing degradation of the device. If you did cause degradation, then you would likely see them, if appropriate to the technique. So again, if it's STEMI, non-volatile, then and you may be able to say by LCMS, you know, as long as for example, if you've got polymer in there and it starts to degrade and that polymer is amenable to the ionization source of choice within LCMS, then you'll start to see it. And all that's telling you is that you need to go and readdress your extraction conditions. But degradation itself is very important outside of the ENL. So it's actually, again, there's more parts within the ISO 10993. Part 9 covers the degradation. I think part 13 to 15 are the different chapters, depending on whether it's polymers, ceramics, or metals and alloys. So there's a lot of information there, but it's a separate process to ENL.
Thank you. Here's another question. We only work with medical grade polymers. Will we still need to perform ENL testing?
I've got that. So the requirement to perform ENL, again, it's based on the risk assessment of building the biological evaluation plan. So as kind of quickly outlined in the early part of the talk, the device class, how it's used, duration of contact. So you need to kind of have these discussions with the toxicologist. It's a bit misleading because medical grade is not a regulated term. So each manufacturer can have different approaches to how they class a medical grade. So on the whole, it does tend to mean that they have greater analytics on the material, providing you, should I say, so you get the compound list that could come from that material, so it helps you with your more targeted, although that's not guaranteed. More commonly, I see it to do with the supply chain consistency and transparency, so guaranteeing that there won't be any formulation changes without plenty of advance notice, which is so important to the medical device industry and less important to some of the other bigger customers of the polymer suppliers. There is actually a proposal. So the Association of German Engineers, VDI, they actually have a proposal to standardize the term medical grade plastics, which would be, again, that's a good direction for the industry to take, I think. So I'd direct you towards, checking what your actual manufacturer stipulates about how they define themselves as medical grade. And I'd also point you towards the VDI documentation. It's a good read.
Thanks, Ben. What was the paper that Rachel mentioned on relative response factors? Rachel, are you there to answer that question?
Yeah, can you hear me okay?
Yes.
So the paper it's by Georgie Labs, and it's in the Journal of Pharmaceutical and Biomedical Analysis and Qualitative Assessment of Extractables from Single-Use Components and the Impact of Reference Standard Selection.
Fantastic. Thank you. Looking at the time here, I think we're just about out of time, so I think we should wrap up. I'd like to thank you, Rachel and Ben, for your excellent talks today and for answering all those questions, and to thank Caitlin for also jumping in to help answer questions for us today. I'd also like to thank everyone in the audience for attending and participating in our event, and I'd like to thank our sponsor, Waters Corporation, for making today's webcast possible. We would like to ask everyone in the audience to participate in a brief survey from our sponsor, and you can see that survey on the right-hand side of your screen. You'll receive an email alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who might have missed today's live event. We hope to see you all next time. Goodbye.